So if you were to just look with the naked eye, right, an IVF culture dish looks, well, it's like a completely empty piece of plastic.
It really does. Just
Yeah.
a tiny flat disc.
Yeah, exactly. A tiny disc with maybe a barely visible smear of liquid across the bottom. But, if you look closer, if you actually mentally zoom in to the microscopic level, you are looking at one of the most actively engineered high-stakes environments in all of modern science.
Oh, absolutely. It's an environment where a single drop of evaporated water or even a microscopic shift in the air you breathe can literally mean the sheer difference between life and death.
Right. And that is exactly what we're getting into today. So for you joining us right now, we want to welcome you to this deep dive into Episode 8 of the Inside the IVF Lab series. This one is titled Embryo Culture, Creating the Right Environment. And we want to invite you into a space that is, it's entirely defined by its stillness.
Stillness is a great way to put it. It's a very slow, deliberate process.
It is. And just to give you a sense of where we are on this specific timeline, in the previous installment, the focus was entirely on the pronuclear check. That pivotal moment of confirming if fertilization actually happened.
Right, checking for those two pronuclei.
Yeah. But now, that singular hurdle is behind us. The zygote has formed. And immediately, like without a second of pause, it faces its next monumental challenge, which is continuing to divide and grow completely within laboratory conditions.
And that is a monumental challenge. I mean, the word invisible is really the perfect place to anchor this discussion.
Okay, let's unpack this then, because our mission here is to reveal that invisible science, right? To the naked eye, that dish looks empty, but it's not.
No, it's not empty at all. And to appreciate what the laboratory is actually doing for that zygote, we have to kind of discard the traditional idea of what an incubator is.
Oh, interesting. How so?
Well, the foundational philosophy of modern embryo culture is that it is never passive storage. The laboratory cannot force an embryo to grow.
Right. You can't just make it happen.
Exactly. There is no chemical additive, no electrical shock, no button to push that will make a cell divide if it doesn't already possess the inherent biological capacity to do so.
Wow. Okay, so if the lab isn't making it grow, what are they doing?
What the lab provides is a pristine, highly engineered, temporary world. The entire goal is to engineer an environment so devoid of stress, so perfectly balanced, that the embryo's intrinsic biological potential can just, safely unfold without wasting any energy fighting against external obstacles.
I really want to linger on that transition for a second. The transition from the zygote to this temporary world. Because the nature of the challenge has completely shifted, hasn't it?
It has, completely.
Up until this point, the challenge was all about access, like the sperm reaching the egg, penetrating the shell. But now it's all about sustained survival in this totally artificial space. So the embryologist sees those two pronuclei. What is the actual physical reality for that single cell in the immediate aftermath?
Well, it is a journey of profound vulnerability, and it's governed by the relentless physics of the cell cycle.
The cell cycle.
Right. Once a normally fertilized zygote is identified, it enters a developmental sequence that operates on a strict biological clock. I mean, over the next 5 to 6 days, it has to progress through very distinct morphological changes.
So it's on a schedule.
Exactly. It begins as that single-cell zygote. Then it enters what we call the cleavage stage. The single cell divides into 2 cells, then 4, then 8.
And wait, when it's cleaving like that, is the whole embryo getting bigger?
No, and that's the critical aspect of the cleavage stage. The embryo does not grow in total size. It is encased in a hard protein shell called the zona pellucida.
Oh, so it's trapped in a shell.
Right. Because it can't expand, the cells are essentially cleaving the existing cytoplasm into smaller and smaller pieces. A single 100-micron cell becomes 2 50-micron cells, and so on.
Wow.
Following this, if it continues to thrive, the cells begin to merge and compact into a tight, featureless ball known as a morula.
A morula.
Yeah. And eventually, it forms a blastocyst, which is this highly complex, expanding structure with hundreds of differentiated cells and a fluid-filled cavity. Orchestrating that sequence requires an immense amount of cellular energy.
It sounds exhausting, honestly. It almost sounds like setting up a very high-tech greenhouse.
A greenhouse, I like that.
Yeah, like if you are an expert gardener, you can meticulously calibrate the UV lights, right? You can balance the nitrogen in the soil, you can dial in the ambient humidity perfectly, but you can't physically reach into the dirt and pull the sprout out of the seed. You can't mechanically force a leaf to unfurl. All you are doing is creating a sanctuary.
Exactly. You are systematically removing every conceivable obstacle so the seed can just execute its own programming.
That's fascinating.
The greenhouse analogy captures the intent perfectly, particularly regarding the removal of obstacles. But in the context of the IVF lab, warmth, for instance, is not about providing comfort.
Oh, it's not. I just assumed it was to keep him cozy.
Not at all. It is about strict cellular protection. You see, the human embryo is biologically programmed to exist deep within the core of the human body, locked at a very precise 37 degrees Celsius.
Right, body temperature.
Yes, and it possesses absolutely no internal mechanism to regulate its own temperature. Inside those cleaving cells, there is a microscopic mechanical structure called the spindle apparatus.
The spindle apparatus. Okay, what does that do?
It is essentially a network of tiny cables that physically pull the chromosomes apart during cell division to ensure each new cell gets the exact correct amount of DNA.
Wow, so they're physically hauling the DNA around.
Exactly. And those microscopic cables are exquisitely sensitive to thermal shifts. If the ambient temperature drops by even a fraction of a degree, those cables can begin to depolymerize.
Wait, depolymerize? Like they break down?
They literally dissolve and fall apart.
Oh my God, just from a fraction of a degree.
Yes, and if the cell attempts to divide while the spindle is compromised, the chromosomes scatter haphazardly. This results in aneuploidy.
That's a genetically abnormal embryo, right?
Exactly, one that cannot result in a healthy pregnancy. So when the laboratory obsesses over maintaining 37 degrees, it is not keeping the embryo cozy. It is maintaining the structural integrity of the embryo's DNA transport system.
That drastically changes how you visualize the physical space of the lab. You aren't just, setting a thermostat on the wall. Every single surface that dish touches has to be contiguous heat.
Every surface, the heated stages under the microscopes, the warming blocks on the counters, all of it.
But if the laboratory is building this flawless greenhouse, the next obvious question for you listening is, well, what exactly is the soil?
The soil?
Yeah, what is this microscopic seed physically sitting in? Because I know it's not just a drop of warm water from a tap.
No, it is certainly not tap water. The liquid environment is known as the culture medium, and it is a highly complex, rigorously quality-controlled chemical solution.
How complex are we talking?
Incredibly. For decades, the evolution of IVF was largely stalled by our inability to formulate a liquid that could keep an embryo alive for more than a couple of days. Early media were really just simple salt solutions.
Wow, just salt water, basically.
Pretty much. But today, culture media are intricate recipes designed to mimic the shifting chemical environment of the human reproductive tract. The liquid provides a foundation of specific salts to maintain cellular balance.
Okay, salts.
It contains buffering systems to manage acidity. It incorporates essential and non-essential amino acids to help the embryo build proteins. And crucially, it contains energy substrates.
Energy substrates, meaning like the fuel for those cellular divisions you mentioned earlier. Because cleaving a cell in half must burn an enormous amount of calories, metaphorically speaking.
Precisely. It burns a lot of energy.
And the embryo's dietary requirements actually change dramatically as it grows.
Wait, they change? Like a baby moving from milk to solid food? Similar concept, yeah. A single-cell zygote, or a two-cell embryo, relies very heavily on a substrate called pyruvate. It processes pyruvate through specific metabolic pathways because its ability to use complex sugars is still dormant.
Ah, so it can't handle sugar yet.
Right. But as that embryo develops into an 8-cell embryo and eventually into a blastocyst with hundreds of cells, its metabolism shifts entirely. It requires massive amounts of glucose to fuel that rapid expansion and the pumping of fluid into its cavity.
So the food literally has to change as the baby grows. That makes total biological sense.
Yeah.
But practically, how do you do that? Does the lab literally go in and feed the embryo a different liquid halfway through the week?
That specific question actually represents one of the major philosophical divides in modern embryology.
Really, a philosophical divide over feeding.
Yes. There are two primary approaches to culture media. The first is what you just described, known as sequential media.
Okay, sequential.
In this approach, the lab uses one specific liquid formulation for days 1 through 3, heavily leaning on pyruvate and specific amino acids to mimic the environment of the fallopian tube.
Because that's where it would naturally be on those days.
Exactly. Then on day 3, the embryologist physically removes the embryo from that drop, washes it, and places it into a completely new dish containing a different medium.
And that new medium is the one with the glucose.
Right. One that is rich in glucose and other factors, mimicking the environment of the uterus.
I mean, that makes sense. But I can hear the implicit risk in that process. because to move the embryo, you have to take it out of the incubator, expose it to the room, physically handle it with a pipette, and move it to a new home. You're interrupting the stillness.
You are, and that is a significant interruption,
Mm-hmm.
which is why there is an alternative.
Which is?
The alternative is the single-step media system. In this approach, the laboratory uses a single, highly complex liquid formulation that contains every single nutrient, amino acid, and energy substrate the embryo could possibly need from day 1 all the way to day 6.
Oh, wow. Like putting the embryo at an all-you-can-eat buffet? It just takes whatever it needs at the moment it needs it.
That is exactly the biological theory behind it. The embryo is an active, intelligent participant in its own metabolism.
That's so cool.
If you provide the full buffet, the embryo's cellular machinery will simply upregulate the transport proteins for pyruvate on day 1, and then later upregulate the transport proteins for glucose on day 4.
It just knows what to do.
Exactly. And the profound advantage of the single-step system is that it eliminates the need to physically move the embryo halfway through its development. You place the zygote in the drop on day 1, and you do not have to disrupt its chemical or physical environment for an entire week.
The single-step approach seems to align perfectly with that philosophy of stillness we were talking about. But regardless of which liquid recipe the lab uses, how does the embryologist actually set up this environment?
What do you mean?
Well, I picture a Petri dish like you see in high school biology, right?
Yeah.
There's a big pool of liquids sloshing around in there.
Oh, no. The physical architecture of the dish is radically different from a standard Petri dish. The embryos do not live in a vast pool of liquid.
They don't.
No. They are cultured in minuscule, precisely measured droplets of medium. We are talking about volumes as small as 10 or 20 microliters.
Wait, how small is a microliter?
Well, to visualize that, a single teardrop is roughly 50 microliters.
50. So these embryos are living in a fraction of a teardrop.
Yes, a fraction of a teardrop.
Okay, I have to pause and push back on that design on behalf of anyone listening right now, because intuitively, that sounds incredibly dangerous.
It does sound precarious.
Right. If you have a microscopic embryo sitting in a droplet that is a fraction of a teardrop, even the tiniest amount of evaporation would be catastrophic. The salts would concentrate, the liquid would turn into like a toxic wasteland.
Yeah.
Wouldn't a massive swimming pool of media be infinitely safer?
This is a very logical question.
If a little water evaporates from a swimming pool, the overall chemistry barely changes. Why force them into these precarious tiny micro droplets?
Your intuition about the risk of evaporation is completely correct. But we use tiny droplets because embryos are not just passively absorbing nutrients from the liquid.
They aren't.
No, they are actively secreting molecules back into it. They produce what we call autocrine and paracrine factors.
Okay, what are those?
These are chemical signals, interleukins, and specialized growth factors that the embryo generates to support its own cellular division and to condition its environment.
Oh, wow. So they are actively changing the liquid around them to make it more hospitable for themselves.
Yes. And if you connect this to the bigger picture, when droplets are tiny, stability is everything. If you place an embryo into a massive swimming pool of culture medium, those precious secreted growth factors simply diffuse away.
Oh, they just get lost in the water.
Exactly. They're instantly diluted into the vastness of the liquid, and the embryo loses the benefit of its own hard work. It is essentially screaming into a void.
That's a sad image, screaming into a void.
It is. But if you confine the embryo to a tiny 10 microliter droplet, those secreted factors become trapped and highly concentrated in the immediate vicinity of the embryo.
I see.
It builds its own localized, highly supportive microenvironment. It is a masterpiece of biological efficiency.
Wow. You are forcing it to live in a tight space so it can build its own chemical aura.
Yeah.
But that brings us right back to my fear about the evaporation. How do you stop a 10 microliter droplet from just vanishing into thin air over the course of 5 days?
This is where a truly critical yet completely invisible piece of laboratory engineering comes into play. The oil overlay.
Oil, like cooking oil.
Medical-grade mineral or paraffin oil. Once the embryologist carefully pipes those tiny droplets of nutrient media onto the bottom of the plastic dish, they immediately flood the entire dish with a thick layer of this oil.
Oh, wow.
The oil completely covers and submerges the water-based droplets.
But the oil acts like a physical lid. It just seals the water in.
It acts as an absolute physical barrier to evaporation. Water cannot pass through the hydrophobic layer of oil, and this is how we maintain the osmolality of the droplet.
Osmolality, let's break that down for the listener.
Sure. Osmolality is the measure of how concentrated the dissolved salts and sugars are in the water. The cell membrane of an embryo is semi-permeable. It allows water to pass through freely to balance the concentrations on either side.
Right, like osmosis from basic biology.
Exactly. If the oil were not there and water evaporated from the droplet would become hyperosmotic, meaning it would have a higher salt concentration than the inside of the embryo.
And water always moves to dilute the saltier side, so the water would actually rush out of the embryo cells into the droplet, causing the embryo to shrivel and collapse.
Exactly. The cells would undergo extreme osmotic stress, shrink, and trigger apoptotic pathways, effectively initiating their own death.
That is terrifying.
It is. The oil overlay prevents this entirely, but it does more than just trap water. The oil also acts as a thermal buffer, slowing down temperature shifts when the dish is briefly removed from the incubator.
Okay, so it holds the heat in a bit longer too.
Yes. And crucially, it acts as a gatekeeper for the dissolved gases that manage the acidity of the liquid.
Okay, here's where it gets really interesting, because that brings us perfectly into the invisible chemistry of the air. We've established the liquid, the soil. We've established the oil that protects it. But that liquid, even sealed under oil, is highly sensitive to the atmosphere around it.
Extremely sensitive.
We've touched on temperature, but let's dive into the pH, the acidity or alkalinity of the droplet. Because you can't just mix a liquid to a perfect pH of 7.3 and expect it to stay there for 5 days, can you?
You absolutely cannot. The biological processes of the embryo itself generate metabolic waste, which naturally alters the pH over time. But more importantly, the culture medium utilizes a specific chemical mechanism called a bicarbonate buffering system to maintain its pH. Bicarbonate acts as a chemical sponge, absorbing excess acid to keep the liquid stable.
Okay, a chemical sponge.
But here is the critical catch. A bicarbonate buffer only functions if it is in constant dynamic equilibrium with carbon dioxide gas.
Okay, wait, break that down for us. Where is the carbon dioxide coming from? Because we breathe out CO2.
In a natural pregnancy, the mother's blood supply provides a constant, rich source of dissolved carbon dioxide to the fluids in the reproductive tract.
Right, okay.
In the laboratory, the incubator must simulate the mother's blood. The incubators are hooked up to pressurized tanks and precisely mix the internal atmosphere to maintain a concentration of roughly 5 to 6% carbon dioxide.
Okay, so they pump CO2 into the box.
Yes. And this CO2 gas permeates down through the oil overlay, dissolves into the microscopic water droplet, interacts with the bicarbonate molecules, and locks the pH of the fluid precisely into the optimal zone for human cellular development.
Wow. That is a terrifying realization. The carbon dioxide isn't just ambient air floating around the box. It is an active structural component of the liquid.
It absolutely is.
The gas is chemically holding the reality of that droplet together, which means, wait, doesn't that mean the regular air in the laboratory room, the air the embryologists are breathing while they walk around, is fundamentally hostile to the embryo?
It is entirely hostile.
Because the air we breathe has barely any carbon dioxide in it, right?
Right. Room air contains about 0.04% carbon dioxide. The incubator contains 6%.
That's a massive difference.
It is. This raises an important question about time. The moment an embryologist opens the incubator door and brings that dish out onto the microscope stage, the laws of physics take over.
What happens?
The high concentration of CO2 dissolved in the droplet immediately begins to off gas. It diffuses out of the water, up through the oil, and escapes into the room air to seek equilibrium.
Because it wants to balance out with the room.
Exactly. And as the CO2 leaves the liquid, the bicarbonate buffering system unravels. The pH of the culture medium begins to rise rapidly, becoming alkaline.
And I'm assuming alkaline is bad.
An alkaline environment is incredibly toxic to the internal proteins and enzymes of the embryo.
How fast does that happen? Because the embryologist has to look at the embryo at some point to know if it's growing. They have to take it out. How do they manage that physical reality?
They manage it by changing their entire perception of time. An embryologist does not think of pH merely as a static number on a readout. They experience pH indirectly as a countdown timer.
A countdown timer. Wow.
They measure it in seconds of exposure. They know that depending on the volume of the droplet and the thickness of the oil, they may have only 90 seconds to 2 minutes outside the incubator before the pH drift crosses a dangerous threshold.
90 seconds. That is intense.
Every action is choreographed. The movement of the dish, the adjustment of the microscope focus, the assessment of the cells. It is a literal race against the chemistry of the room air.
That level of discipline is staggering, measuring acidity in seconds. It's like holding your breath underwater. You know precisely how long you have before the environment becomes incompatible with life.
That's a very visceral way to put it, but accurate.
And the CO2 is just one part of the gas equation, right?
Mhm.
We also have oxygen. Now, we as humans require massive amounts of oxygen to survive. But these culture systems actually suppress oxygen, don't they?
They do. The air we breathe is approximately 21% oxygen. For a long time, early IVF incubators simply used room air mixed with CO2.
Right, because why wouldn't you? We need oxygen.
Exactly. But developmental biologists realized that deep within the human female reproductive tract, specifically within the lumen of the fallopian tubes in the uterus, the environment is actually hypoxic.
Hypoxic, meaning low oxygen.
Yes. The oxygen concentration is remarkably low, typically ranging from 2 to 8%.
Why would the human body intentionally deprive its own reproductive tract of oxygen? That seems counterintuitive.
It is actually an evolutionary defense mechanism. Oxygen is fundamentally a highly reactive, volatile element.
Really? Volatile?
Yes. When cells metabolize oxygen to create energy, they naturally produce byproducts called reactive oxygen species, or free radicals.
Oh, free radicals. I've heard of those. Like in skincare commercials.
Right. They are unstable molecules that strip electrons from other structures. In an adult, we have robust defense mechanisms to handle them, but an early embryo, particularly during the cleavage stages, has very limited antioxidant defenses.
So it can't fight them off.
No. If you expose a day 2 embryo to 21% atmospheric oxygen, you induce massive oxidative stress. Those free radicals begin damaging the lipid membranes of the cells, denaturing vital proteins, and most critically causing fragmentation in the embryo's DNA.
Wow, so atmospheric oxygen literally rusts the embryo from the inside out.
That is a very apt way to describe oxidative damage, yes.
So how do you stop that?
To prevent this, modern IVF laboratories utilize specialized incubators that pipe in pure nitrogen gas. The nitrogen displaces the oxygen in the chamber, forcing the ambient oxygen levels down to a safe 5%, perfectly replicating the hypoxic sanctuary of the fallopian tube.
When you lay it all out like this, the continuous heat to protect the spindles, the heavy CO2 to lock the pH, the nitrogen gas to suppress the oxygen and prevent DNA damage, the oil to stop evaporation. I mean, the sheer logistics of maintaining all of this simultaneously are overwhelming.
It is a profound balancing act.
You aren't just putting a dish on a shelf. The environment requires a machine. So let's talk about the hardware that actually performs this magic, the incubator systems themselves.
The modern incubator is arguably the most critical piece of hardware in the laboratory. Calling it a warm box just fails to capture its complexity.
I can imagine.
It is a highly sophisticated, precision-controlled life support system. It contains sensitive thermal probes, infrared sensors to continuously monitor the carbon dioxide levels, zirconium dioxide sensors for the oxygen, and intricate valve systems that constantly inject microbursts of gas to maintain the absolute perfect atmospheric ratio.
Microbursts.
Yeah.
Just tiny adjustments all day long.
Yes. But the true test of an incubator's quality is a metric we refer to as rapid recovery.
Rapid recovery. Because every time the human element intervenes, the machine has to compensate, right?
Right.
If I'm baking a delicate souffle in the oven and I crack the door to peek inside, all the ambient heat rushes out. The oven takes time to get back to temperature, and the souffle collapses.
I love that analogy. It translates perfectly to the lab, but the stakes here are cellular. The incubator is the center of embryo culture, but not all incubators shape culture in the same way.
How so?
Well, when an embryologist opens the door of an incubator, the perfect, carefully balanced atmosphere of 37-degree heat, 6% CO2, and 5% oxygen instantly cascades out into the room.
Just gone in a flash.
Simultaneously, the cold 21% oxygen room air rushes in. The carefully constructed microenvironment is obliterated in less than a second.
Wow.
Rapid recovery refers to how fast the incubator's internal sensors detect that catastrophic loss, trigger the internal heaters, and open the gas valves to purge the room air and recreate the perfect environment once the door is sealed closed.
And I imagine the speed of that recovery drastically affects the embryo sitting inside.
It dictates their level of physiological stress. Older, large-capacity box incubators had massive doors. Opening the door exposed dozens of dishes at once.
Oh, like opening a giant refrigerator.
Yes. Once closed, those large chambers might take 15, 20, or even 30 minutes to fully recover their precise gas concentrations and temperature. That means the embryos inside were sitting in suboptimal conditions for half an hour just because someone checked on a different patient's dish.
That sounds incredibly disruptive. How did the field solve that engineering problem?
By shrinking the space, the industry evolved toward benchtop incubators.
Benchtop, like they sit on the counter.
Yes. Instead of a large, refrigerator-sized box, a benchtop incubator looks more like a series of very small, independently heated drawers. Each individual patient's dishes are placed into their own tiny, isolated drawer.
Oh, that makes sense.
When the embryologist needs to observe patient A's embryos, they open only that specific small drawer. The microenvironments of patient B, C, and D remain completely undisturbed.
That's brilliant.
Furthermore, because the physical volume of air inside that single drawer is so small, the rapid recovery time when the drawer is closed drops from 30 minutes down to less than 2 minutes. The gas purges almost instantly.
So it isolates the risk and minimizes the recovery time. But, even with a tiny drawer, you still have to open it.
You do.
The embryologist still has to take the dish out, walk it over to a microscope, look through the eyepieces, write down their notes, and walk it back. That countdown timer of the pH drift is still ticking. Is there a way to eliminate that physical movement entirely?
There is, and it represents one of the most profound technological leaps in the history of IVF. It is known as the time-lapse incubator.
Time-lapse, like taking a video.
Exactly. This technology fundamentally solves the ultimate paradox of embryology, which is the desperate need to observe the biology, clashing with the absolute necessity of leaving the biology undisturbed. A time-lapse incubator is essentially an advanced benchtop incubator with a high-resolution microscope and a digital camera system integrated directly into the floor or ceiling of the heated chamber.
So the camera lives inside the box with the embryos.
Exactly. The dishes are placed into the machine on day 1 and the door is sealed. It is not opened again for 5 to 6 days.
Not at all.
Not at all. The internal camera operates on a motorized track. Every 10 to 15 minutes, 24 hours a day, the camera silently slides under each individual embryo. Because an embryo is a three-dimensional sphere, the camera quickly snaps a picture at 7 or more different focal depths, then moves to the next embryo. It uses very brief, low-intensity red light to avoid any phototoxicity to the cells. The system's software then stitches these thousands of microscopic images together into a continuous, high-definition time-lapse video.
That is awe-inspiring. You have a constant security camera feed of the microscopic greenhouse. The embryologist can just sit at a computer monitor across the room, drinking a cup of coffee, and watch the cells divide in real time without ever once letting the cold room air touch the dish.
They get infinitely more data. They can see the exact minute a cell divides, which is a science known as morphokinetics. And simultaneously, they provide the embryo with an utterly undisturbed physical and chemical environment. It is the realization of the perfect sanctuary.
That is incredible.
But it is vital to remember that technology alone does not guarantee success. The hardware is only as effective as the human protocols that govern its use, which brings us to the operational philosophies of the laboratory.
Right. How the humans actually choose to arrange the embryos inside these machines. This moves us perfectly into the philosophy of restraint and how labs choose to culture their patients. Because one of the major decisions a lab makes is between group culture and individual culture.
Yes, that's a key distinction.
If I am a patient, I might naturally assume my 10 embryos are all just hanging out together in one dish. But that isn't always the case. What is the argument for isolating every single embryo into its own private drop of liquid?
Well, individual culture is deeply rooted in the necessity for absolute tracking and data correlation. If you place one embryo into one specific microdroplet and you map that droplet to a numbered grid on the bottom of the dish, you establish an unbreakable chain of identity.
You know exactly who is who.
Exactly. You know with absolute certainty that the embryo in drop 4 on day 1 is the exact same embryo in drop 4 on day 5. This becomes non-negotiable for workflows that involve genetic testing.
Oh, right, because you test them later.
Yes. If you are going to biopsy an embryo, remove a few microscopic cells from its outer layer sent to a genetics lab, you cannot have any ambiguity about which biopsy belongs to which embryo. Individual culture allows the lab to build a highly precise, isolated dossier on every single embryo.
It's a system designed for clean data and zero mix-ups. But then there is the alternative, group culture. This is where multiple embryos from the same patient share a single larger droplet of media.
Correct.
And earlier you hinted at something incredible when we talked about the oil overlay. You mentioned that embryos secrete growth factors into the fluid. If they're in a group, are they actually sharing those factors with each other?
Yes, they are. And this taps into a beautiful and highly complex biological reality. In vivo, inside the human body, an embryo is never truly alone in a sterile void. It is in constant chemical dialogue with the maternal tissues, and if there are multiple ovulations, with other embryos.
So they're communicating.
In a laboratory dish, if you culture 4 embryos together in a 50 microliter drop of media, they share the same fluid space. As they grow and metabolize, they secrete autocrine and paracrine factors.
Right, the interleukins you mentioned.
Yes, an embryo that is robust, dividing on time, and thriving will produce a high volume of these positive signaling molecules. In a shared microenvironment, that thriving embryo effectively saturates the liquid with growth factors, and its neighboring embryos can absorb and utilize them.
That is almost difficult to comprehend. The idea of microscopic cellular teamwork happening inside a plastic dish. Like, hey, I have extra interleukins, you can use some to help your cleavage stage. It makes the liquid feel so much more alive.
It highlights how incredibly dynamic the culture environment is. Many laboratories utilize group culture specifically because they believe this shared chemical communication leads to slightly higher overall blastocyst formation rates. It mimics a more communal, biologically supportive ecosystem.
That's amazing. But whether a laboratory utilizes group culture, individual culture, sequential media, or single step, the overarching human philosophy that dictates their success remains identical, right? The relentless discipline of minimizing disturbance.
Good embryo culture is often defined as much by what the lab avoids doing as by what it actively does.
So what does this all mean for the lab worker? If I'm an embryologist holding the potential future child of a patient in my hands, my instinct would be to constantly check on them, to hover, to make sure they are okay. How do you fight that profound human urge?
It requires a fundamental rewiring of your instincts as a medical professional. In almost any other branch of medicine, care is defined by action. You monitor the vitals, you lay hands on the patient, you intervene.
Right, you actively do things to help them.
In embryology, you must internalize the harsh reality that your gaze is inherently toxic.
Wow, your gaze is toxic.
Yes. Your observation disrupts the temperature, breaks the gas equilibrium, and risks osmolality shifts. Therefore, a great embryologist defines their care by their restraint. They force themselves to walk away from the incubator. They limit their observations to strictly mathematically and biologically necessary checkpoints.
Like when?
Perhaps a brief assessment on day 3 to check cleavage rates, and then nothing until day 5.
It makes me think of a pit crew in a Formula One race. The car is out on the track performing. You don't pull the car into the pit box just to look at it and say, yep, looks good. You only pull it in when it is absolutely necessary.
That's a great comparison.
And when you do pull it in, the entire crew is already in position. The tires are unwrapped, the air guns are spinning, every movement is choreographed. You execute the change in 3 seconds, and you get the car back out into its optimal environment.
The pit crew analogy is astonishingly accurate for a high-functioning IVF laboratory. The physical choreography of an embryologist is a marvel to watch.
I bet it is.
Long before they even touch the handle of the incubator door, the preparation is complete. The heated stage on the microscope is verified at exactly 37 degrees. The lighting is adjusted. The patient's physical chart or digital file is open and ready.
They aren't fumbling around for a pen.
Never. The sterile pipettes are positioned in their hands. They open the incubator, extract the dish, place it on the stage, sweep their eyes over the embryos, mentally register the cell counts, symmetry, and fragmentation in a matter of seconds, and return the dish to the sanctuary before the pH has a chance to drift.
That's unbelievable.
The restraint required to do nothing for 3 days, followed by the intense, high-speed execution of an assessment, is the hallmark of mastery in this field.
That paints such a vivid picture of the sheer physical tension in the room. The silence, the speed, the precision. But we have to talk about the reality of biology, because what happens when the biology doesn't cooperate? What happens when, despite the perfect single-step media, the flawlessly washed oil overlay, the rapid recovery incubators, the hypoxic nitrogen gas, and the perfectly choreographed pit stop assessment things still stall? Because this isn't a fairy tale, this is human reproduction. Let's look at the vulnerabilities and the hard limits of what this incredible environment can actually accomplish.
This is perhaps the most sobering but necessary part of our discussion, because the environment we've described is a masterpiece, but it is deeply fragile.
Fragile in what way?
The reality of the laboratory is unrelenting vigilance, because microscopic failures compound into macroscopic tragedies. If a single heating element in a warm block degrades and the temperature drifts by 1 degree,
Just 1 degree.
or if the microscopic valve regulating the CO2 tank sticks for an hour and the pressure drops, if a batch of mineral oil was not properly washed and retains trace amounts of volatile organic compounds, or if a junior embryologist is inconsistent in their handling speed and leaves the dish exposed to room air for 3 minutes instead of 1,
All of those little things add up.
the embryos will suffer. They will experience metabolic shock, their development will slow, or they will arrest completely.
I cannot fathom the emotional weight that places on the laboratory staff. Because if an embryo stops growing, and you are the god of that tiny universe controlling the air and the water, your immediate human reaction has to be, what did I do? Did I leave the door open too long? Was the media not equilibrated properly? The burden of perfection must be agonizing.
It is a profound psychological burden. Embryologists are acutely aware that they hold the physical manifestation of a patient's hopes in their hands. When development stalls across a cohort of embryos, the laboratory must immediately launch an exhaustive audit of their own systems.
Checking everything.
Yes, they check the pH logs, the gas tank pressures, the lot numbers of the media, the calibration of the temperature probes. They look for any environmental deviation. But this brings us to a crucial series of myths that we must dismantle. Unpacking these myths helps reset expectations, because the environment is only half of the equation.
Okay, let's go through them. The first myth, which we've thoroughly destroyed today, is that culture is just waiting. That once the dish goes into the box, it's just a waiting game. Clearly, it is a highly active, constantly maintained state of chemical engineering. But the second myth is that the incubator alone is what grows the embryos.
Yes, the incubator is merely the engine block. The success of the culture is the entire interconnected web. The specific formulation of the media, the physics of the oil, the purity of the gas, the speed of the human hand, and crucially, the intrinsic quality of the embryo itself.
Right. Okay, myth number 3, more checking is better. If the patient asks, can you look at them every day to make sure they are okay?
As we explored with the philosophy of restraint, in the absence of a closed time-lapse system, excess handling is actively detrimental. Looking closer does not improve the biology.
It makes it worse.
Yes, the physical act of observing introduces thermal and chemical stress that hinders development. Sometimes the most profound care is to close the door and walk away.
And finally, the fourth myth, which speaks directly to that agonizing emotional burden we just discussed. The myth that poor embryo development always means a bad culture system. If a patient's embryos don't make it to the blastocyst stage, the natural assumption is that the lab must have messed up the environment.
This is the harshest reality to convey to both patients and the laboratory staff themselves. The laboratory can control the environment with absolute flawless precision, but the laboratory does not and cannot control the embryo's inherent genetic blueprint or its biological energy reserves.
The genetics are already set.
Yes. If an embryo possesses severe, complex chromosomal abnormalities from the moment of fertilization, or if it was formed from an oocyte with depleted mitochondria that cannot generate enough ATP for the cleavage stage, that embryo is not going to develop into a healthy blastocyst.
No matter what the lab does.
It does not matter if the temperature was perfectly locked at 37 degrees. It does not matter if the pH was an unyielding 7.3. The environment can only support the potential that already exists within the cell. It cannot manufacture potential out of thin air.
So to synthesize everything we have explored today, to pull all these incredibly dense microscopic details together, embryo culture really is the exact intersection where human developmental biology meets high-level environmental engineering.
That's a perfect summary.
It is a partnership. The laboratory builds the absolute perfect stage. They set the precise lighting, they perfectly tune the acoustics, and they mandate absolute silence from the audience. But the embryo still has to walk out onto that stage and perform the play. And sometimes the biology simply cannot bridge the gap, even when the environment is flawless.
That is the beautiful, tragic, and miraculous reality of IVF. Understanding the extreme limits of what the lab can control is vital. It lifts the unfair burden of absolute omnipotence off the shoulders of the embryologists, and it grounds the expectations of the patients. But simultaneously, understanding this immense complexity highlights something extraordinary. When you realize how incredibly narrow the parameters for cellular survival are, how the temperature, the osmolality, the buffering systems, and the gas pressures all have to align with the embryo's shifting metabolic needs.
It's staggering.
It is. Every single blastocyst that successfully forms in that gystic dish is an absolute testament to human ingenuity and the sheer raw resilience of biology.
It is a miracle of logistics meeting life. And that brings us to the close of this incredibly detailed journey through episode 8 of the source material. Let's take a moment to recap the invisible world we've uncovered today.
We've covered a lot of ground.
We really have. We started with the vulnerable, newly fertilized zygote and followed it into the complex liquid world of culture media. We examined the shifting metabolic needs from pyruvate to glucose and the debate between sequential and single-step environments. We explored the vital physics of the oil overlay, protecting the tiny microdroplets from evaporation and osmotic shock. We dove into the invisible chemistry of the air, the continuous heat protecting the cellular spindles, the precise injection of carbon dioxide to lock the bicarbonate buffer and manage the pH, and the suppression of oxygen to prevent free radical damage.
The environment really is everything.
It is. We looked at the hardware, the wombs of the lab, understanding the critical nature of rapid gas recovery and the revolutionary impact of time-lapse video incubators. We discussed the agonizing human philosophy of restraint, the microscopic communication in group culture, and the choreographed speed required to minimize disturbance. And finally, we faced the hard truth of the lab's limits, that the perfect environment can only support, never create, intrinsic biological potential.
It is a staggering sequence of interconnected dependencies, and it perfectly sets the stage for our next deep dive.
Yes, it does, because in episode 9, we're going to pivot our focus. We will transition from looking at the environment to looking directly at the embryos themselves.
That's going to be an exciting one.
Next time, we will dive deeply into cleavage stage development. We will explore exactly what those embryologists are searching for during those high-speed pit stop assessments on days 2 and 3. We will break down how they grade cell numbers, assess cellular symmetry, and identify cytoplasmic fragmentation, and we will reveal how those very first cellular divisions begin to tell the story of how the embryo is responding to the meticulous environment we just spent this entire time describing.
It is going to be incredibly fascinating to finally see the biological results of all this invisible engineering.
I can't wait. Thank you for joining us on this slow, deliberate exploration of the laboratory's most invisible yet most foundational phase. We hope taking this time has given you a profound new appreciation for the silent, exacting, and deeply compassionate work happening inside those incubators every single hour of the day.
It really is a labor of love.
It is. And as we leave you, we want to offer a final thought to mull over on your own. As we've extensively explored today, an embryo in an IVF dish is profoundly, unyieldingly dependent on its environment.
Completely dependent.
Its very DNA is protected or damaged by the exact temperature, the specific dissolved gases, and the delicate balance of the liquid it sits in. But isn't that fundamental truth applicable far beyond the laboratory?
That's a beautiful way to think about it.
Right. From a 4-cell embryo floating in a microscopic droplet to a fully grown human being navigating the world, we are constantly, silently shaped by the invisible environments that surround us. We spend so much of our time obsessed with the seed, our DNA, our inherent talents, our intrinsic nature, that we so easily forget how utterly and fundamentally dependent we are on the soil we are planted in and the air we are given to breathe.
It's so true.
The microscopic embryo fighting to divide in the stillness of the IVF dish isn't an exception to the rule of life. It's just the most distilled microscopic proof of that universal truth. Until next time.